Introduce the plenary speaker, Nicole Soranzo. She is a fantastic colleague at the Human Technopole, and she has been covering several roles and many important positions. She was a senior group leader at the Sang, professor in Cambridge, and many more achievements. And also she is a fantastic colleague at the Human Technopole, where she is head of research center of the genome, so IMI, so we are sharing a similar, co-heading the genomics center, and we have very good science discussing friend at the Human Technopole. Nicole, probably we can go ahead and leave you your microphone slides. Get away.
Thank you, Piero. So, all right. So, my personal disclaimer: I'm a human geneticist, but what I really want to give you is a different perspective of how we're thinking about using single-cell data to address some fundamental questions in human genetics. And mostly, I don't want to talk about past career, but rather focus on a generation of a data set that started with my group at the Sanger Institute and is continuing in the analysis from Human Technopole. The timer hasn't started. I don't know if I'm aware this is the last talk of the day. So really, thank you very much for this invite. So we're really interested in understanding or finding new paradigms to try and tackle the genetic basis, but also the environmental basis of complex diseases. Complex diseases are, cardiovascular diseases, cancers, you name it. They are the biggest, of course, killers, so a massive health problem. But importantly, they are also growing in prevalence dramatically due to changes in environments. And I've been very lucky to be part in the last 15 years or so to be part of the human genetics community at the stage where we're beginning to reap the fruits of the human genome sequencing and essentially developing approaches to study complex traits using genome-wide association studies and genome sequencing. And this is, if you're interested in the topic, a very nice paper, a review paper led by Mark McCarthy here in the audience that really recaps fundamental themes and advancements that we made in the field. And this has really been through a combination of three factors, a virtuous cycle of three factors, the creation of large-scale population resources that have been enabled by the development of highly performing genomic technologies, so that have in time allowed to scale the insights that we could get from the genome, and in turn, development of a knowledge base driven from these large-scale data sets. And so when I started working in complex traits, we knew very little about genetic predisposing loci. And in the last 10 years or so, we discovered hundreds of thousands of loci in the human genome that control predisposition to complex traits. And we've been very busy, and in fact, this continues to be the case, in continue to characterize through new discoveries what are the unique genetic factors implicated in disease risk, and particularly how these interact with environments, through various types of genomic technologies to dig very deep into functional consequences of genetic variants, and particularly to understanding mechanisms that drive unique disease predisposition. And this is important because this type of data is fundamentally important to advance or accelerate the pace of genomic discoveries of targets for treatment, as well as for tools for improving the prognosis and diagnosis of diseases. And why is it so hard to study complex diseases? Because, in fact, these diseases are very complex. And so if you think about diseases like obesity or type diabetes, they really are the combination of a variety of very complex molecular mechanisms that reflect both intrinsic set points determined by genetics, biological factors, age is clearly one of them, but not only, but also environmental factors, stressors, for instance, the presence of an infection at a given point in time or even in the past, history of the person, or environmental effects like stress, pollution, etc. And finally, of course, also lifestyle variables. And so all of these contribute in a way that is complex, often non-linear, and typically when we study disease in isolation, it's very difficult to capture the complexity of this relationship and interactions. So this really calls for different paradigms that go beyond the single variant, single gene, single disease. And really there are key points that I think can drive these new paradigms. One is to try and find ways to achieve better or fine-grained assessment of disease states in different people, to model more comprehensively real-life scenarios where all these complex factors interact, to develop models, both statistical but also data-driven, that allow you to assess the joint contribution of biology, but also genetics, exposure, and comorbidity in real-life settings. And finally, and very importantly, is the ability to jump back and forth in biological scales to bridge molecular mechanism with all the way to population risk. So as I said, we've been very lucky to work with population cohorts. I will not tell you too much about that, but the fundamental characteristics of this is typically they collect individuals that are healthy or predominantly healthy. They might have extensive ranges of phenotypes collected to them. They capture environments and disease. And I just want to mention two aspects that are equally important. One is size. In order to do this statistical inference properly, we need large sample sizes. And UK Biobank has been the premier cohort for this. It's used all over the world. And the other is the ability to tap broadly into the diversity, not only the genetic predisposition, but also of environmental factors. And so, for instance, in a cohort in the UK called Genes and Health is a cohort of British, South Asian individuals of Pakistani and Bangladeshi origin that live in the UK, but experience very different rates of environmental exposure. They have different practices of access to healthcare, and also, indeed, they are quite divergent genetically, particularly due to excess of homozygosity. So we can take these differences actually into strengths, and we can use an ability to compare and contrast information from these two types of studies to understand disease better. So I, in my role at Sanger Institute, I worked a lot in defining essentially intermediate phenotypes that allow us in population cohorts to bridge the space between genetics and disease risk, and we have explored over the course of many years all layers of information. And most of this kind of information was derived from circulating plasma and blood, and this information that proved extremely useful, but often not sufficiently detailed. So they are coarse measurement of variable phenotypes. So the design typically we use is this assessment of baseline traits, where essentially, as I told you before, we use locus discovery based on molecular phenotypes and a triangulation of effects across data layers. We're now beginning to look prospectively, so to take essentially from health records a disease assessment over time and try and use this information, a baseline, to predict the long-term risk of disease. But really what we are interested in is defining completely different paradigms of analyzing the data, and really thinking about human population as experimental systems. And so, in a way, revert to this kind of relationship, canonical relationship of measurement. And so, this is really what we are testing now. So, let's think about human population as a complex experimental system. We can use this rich phenotypic data that comes from baseline data, but also the rich phenotypic data in cohorts that come from questionnaire, from health records, from objective measurement of physical activity, etc., as essentially exposure to the system. And then we ask, can we have a molecular readout of this exposure that allows us to treat a human population essentially as an in vivo perturbation system? And this is really where this project idea came from. So of course, we want to have fine-grained resolution of this type of effects. And so we put forward, together with many colleagues, the vision of so we exploited the fact that single-cell technologies were becoming more widely available and cheaper to begin to think whether we could apply this paradigm of population-scale profiling at single-cell level. And so this was the vision. And we were really lucky that we had a fantastic relationship with John Akul, who agreed to fund a special project from Transaction Formation that allowed us to do this project. So my co-PI initially were Sarah and Oliver, that you all know, but we also partnered later with David Mahill and Hilary Martin to include the Genes and Health cohort. And there are many, many colleagues that now joined this project. So this is called Cardinal, and it was supposed to be a pilot project for a larger scale exploration. So what we decided to do is to say, okay, let's take blood from individuals from deeply phenotyped cohorts where we have access to genetic data, but also to multi-omic modality, imaging, environmental exposures, mortality from health records, and multiple clinical phenotypes. And let's agree to profile single cell from circulating peripheral blood mononuclear cells at about a rate of between 1,000 and 2,000 cells per person as a way to create essentially a molecular fingerprint for the health status of that person. And we had a number of goals in mind. Of course, the first goal was to obtain a deep characterization of cell types, but also states, and hopefully in the future also programs, and also compositional phenotypes. So for instance, the ratio between different types of cells at an unprecedented resolution. We're of course interested in genetics, so we want to create expansive atlases of genetic effects, and we want to use these atlases to study the genetic contribution to complex diseases through the single cell lens. And it was very important for us that this big effort would result in a resource for the broad community, and this really drove the choice of the cohorts that we picked. So I just want to take you a little bit through also the data production aspects of it. I'll go a little bit fast. There's a lot of detail, but I just brought it out there because if there's anything you also would like to follow up on, I'll be very happy to do that. So of course, we want to do a massive data generation effort at scale. And this requires, first of all, automated error-free processes, particularly because of the accountability of this large-scale cohort they have to patients and to the global community. We wanted to develop robust experimental pipelines, to develop automated analytics and diagnostics that would allow us to follow the data production in real time. And of course, a big concern is the ability to access biological sample in the thousands of people in a limited amount of time. So we took advantage initially of the fact that UK Biobank, so UK Biobank, as I said, is the largest cohort in the UK. It's 500,000 volunteers that have been now profiled, an extensive number of phenotypes. They are predominantly from white British background, quite middle class. These individuals were first enrolled in the study between 2006 and 2010. So we now have about 20 years of follow-up on their health status. So there's now a big effort to recall 100,000 of these 500,000 people to do full-body MRI imaging at high resolution. And so we took advantage of this recall to essentially ask UK Biobank, would you give us an aliquot of blood for every person that goes to the recall? Of course, we couldn't do 100,000 people, but we settled on 5,000 individuals as a proof of concept. And as I remind you, there is both a lot of baseline data, imaging data at the time point of blood collection, and also information on health records throughout history. The second cohort, as I said, is genes and health. So this was really prompted by Jonah's desire to increase the diversity of our study. And here, this is a more recent study. The baseline collection happened only a few years ago. There is not a lot of omic data, but there is linkage to electronic health records. And the difference between this is that, as I said, these are populations that experience very different rates of health, of disease, because of cultural practices, a little bit of cultural isolation within the British society, but also and tend to access healthcare less than the average white British person. And so in this case, we collected the blood from 1,500 people from an active community-based recall. So the recall was done essentially for us. And in addition to single-cell data, we also profiled multiple metabolomic and proteomic data dimensions. So I'll go very quickly through the operational considerations, but I just want to give you a sense of what putting together a project of this scale entails. So essentially every day, across 5 different recruitment centers in the UK, blood was drawn, and the blood was collected before 5 o'clock in the evening, and then it was flown or driven overnight to the Sanger Institute. The blood was collected by our sample receipt team, with Samina and Laura primarily. We immediately went to the lab. We did a hematology analyzer analysis, and then we set up, as I said, an automated process for single cell collection. And down the line, we've been really busy in understanding how to process, QC, and analyze the data. So the design was really driven by the need to make the experiment cost-effective, but also the practical constraints. So we essentially settled on a design where 12 samples were mixed. The blood of 12 people were mixed. In fact, the PBMC were mixed in a single pool. And then down the line, we would deconvolute the cells from each donor through genotype-based fingerprinting. And there was a lot of consideration in designing what was the optimal study design, but we picked a design that was feasible given the amount of money that we had, but also that was the most cost-effective. And I really want to credit Stephen Watt for driving the process of setting up all this experimental pipeline. And so what we do then, we have a completely, other than the initial point of collecting blood into the lab, everything else is completely hands-free. So we set up a first step of immune cell purification on a Hamilton robot. So from this case, we were able to extract essentially through monomagnetic isolation about 150,000 PBMCs from 150 microliter of blood. And this allows to process about 100 samples per day in a single experiment. We then took the PBMCs resulting from this step. We counted them on a cell plate-based reader in order to create equal pools of equal number of cells for donors. And these donors were essentially rearrayed on a Hamilton machine before the 10x reaction that was done through the 10x Genomics Connect instrument. And so here we load about 40,000 cells per channel to retrieve 25,000, and that gives us about 2,000 cells per donor. So this is the kit. This is the extensive benchmarking that we did to make sure that the processes were calibrated and worked as we expected. And just to say this, of course, we need LIMS to make it robust. And I have a video. I won't play all of the video, but I just want to flag the fact that in addition to the single cell, we also collected Sysmex hematology data. And that's really important because it allows us to move from just looking at cell proportion within a unit of PBMCs to actually absolute cell count. And this is very important for analysis. I'll go really, really quickly about this, but essentially we have now designed very extensive data processing workflows that allow us to process the data in a high-throughput manner, and I'll take you quickly through the steps. So as I said, we sequence 10 to 12 individuals per pool. We sequence 8 pools per day in sequencing tranches. This goes straight to the sequencers. The data comes off the sequencer and goes through an automated pipeline that is called YASCP, and this is now publicly available, was developed mostly by Mathis Ozol, who is here together with many others. And so this automatic pipeline takes the data in real time, aligns the barcode, removes ambient RNA, deconvolutes donors, and does automated cell annotation, double marking, and clustering of the data. And so we can process about 120 samples within 30 hours, like straight away. We then do quite a lot of manual work to make sure that the data is completely correct. So we go through the lab metadata, we make sure there is no sample swaps, everything is annotated, and essentially this results in what we call usable cell and usable barcodes that define the QC pass data set. In addition to this, we also, of course, do a lot of other downstream filtering. I don't really want to say to that, but we spent actually an incredible amount of time trying to think about what are the self-filtering and the normalization and aggregation steps that preserves the most data without compromising the quality of the data. And just to give you an example, this is unreadable, but just to say these are cell types from the rarest to the most common, and from 0 to 1%, this is how much we remove using different filters. And so as you see, some types of filters are incredibly punishing and remove all the rare cell types from our population. So there's a lot of lessons to be learned in dealing with a data set of this type. Finally, we take the data, we put them through integration pipelines. You all know very well what they are. But essentially, this integration pipeline allows us to remove the effect of known confounders. And one principal one is the sequencing batch. We have 300 sequencing libraries. And so the data that comes out of the other end is very high quality. And I'm very happy to talk about any of this later on. So going to a little bit more the biology, what do we see in this type of data? So this is essentially the downstream to this annotated pipeline annotation effort. So we take essentially the output of annotated approaches such as cell typist, azimuth, and cell hint to essentially curate the quality of the cell populations that we can see from our data. This is a result of the integration. As you see, the two cohorts are perfectly overlapping. There is absolutely no evidence for batch effects. And indeed, even after manual curation, we can use approaches such as CRCs to transfer labels across populations, and that works really well. So in the current freeze that we're working on, we have about 7 million cells in the UK Biobank and about 2.5 million cells in the Genes and Health cohort. So what do we see? We see essentially a high-resolution view of all the primary immune cell types. So there's 28 common cell types annotated in this data. There's about 4.5 million T cells, all combined with 18 different types in UK Biobank, and about 1.5 million cells in Genes and Health. There is nearly 1 million B cells across the two cohorts, split across 7 cell types, about 2 million monocytes and dendritic cells, and monocytes and dendritic cells. So we can begin to study property of this data and understand how well we've done in capturing not just the very common cell types, but also the rare one. And so what you see here essentially is that if you plot the number of cells per cell type across the number of donors that carry the cell type, you see that there are multiple cell populations that are essentially complete. We see cells in every donor of the study. As you move towards the rare cell types, you see that fewer donors have fewer cells, and this is simply a matter of the sampling coverage. But you also begin to see some interesting differences. So this is the UK Biobank and Genes and Health. For instance, UK Biobank has a very high prevalence of a population, which we went on to annotate as a putative B premalignant cell type. So this is shown here in the cell label transfer. So about 2% of UK Biobank individuals have this cell type population, but only 0.2% of Genes and Health. And we think this is a signature of a premalignant B-cell condition leading to CLL that we see in UK Biobank, because UK Biobank individuals are on average older than Genes and Health. And we also ask whether this type of data, where we have essentially population-level view, agnostic of disease diagnosis, whether we can begin to identify rare cell types that are indicative of cell states that reflect immune differences. And so this is what we indeed found. So these plots show 3 types of signatures that are essentially B and T cell and NK cells that show signature in interferon response. So these are cells that are, for the most part, absent from the population. So about every person on average carries 1 or 2, but indeed there are individuals that carry hundreds of them. And we can show that this effect is not driven by cell compositional differences between people, but they are real biological signatures. And we went, in fact, back to the health records and found out that these people had been at the doctor within the last week complaining of sore throat or diseases. And so this really suggests, and there's a second set of population that are immune-activated cells showing the same effect. And so this is really a good demonstration that we can now use population-level agnostic approaches to identify rare cell states associated with disease or, if you want, immune perturbations. We also, of course, want to understand disease signatures for complex diseases more broadly, and also with biology. And so you will know, for instance, that immune cells change over the lifetime. This has been shown in atlases such as the Healthy Immune Atlas. And so we wanted to see whether the kind of signatures that are generated in this very controlled experiment can be recapitulated in real data. And the short answer of this is that, yeah, we do very strong effects of a certain subset of both ethnic dimension, sex differences, and also age differences. And I want to highlight the fact that indeed having absolute counts as well as differential counts really provides accurate interpretation of differences that might be driven by skews in ratio. And so this is just, for instance, this is essentially, what we can do now is use this data to model essentially longitudinally the changes in cells, proportions that happen over the human lifetime, and we can accurately predict cells that are increasing or decreasing during the age of the individuals. And this is important because once we can see this effect, we can also model them together with the risk of disease. We also ask whether if we take common conditions that are again inferred from health records, are we able to identify compositional signatures, again from single cell data. So just to remind you, Genes and Health was sampled at this point, but we have health information spanning 20 years or more back. And so we can essentially classify each person in our population based on whether they have a past diagnosis of disease. And this diagnosis can stretch quite a lot back in time and can lead, in fact, in many cases, can be classified not just as transient perturbation, but indeed as chronic differences between individuals. And I'll go very quickly through that, but just to say that indeed we find very strong signatures of cell compositional skews that are associated to a variety of disease diagnoses. This is done in a small number of people. We are replicating them in UK Biobank. So we see signatures of essentially infectious diseases present in the population, of endocrine function disorders. We see signatures of circulatory defects, digestive problem, respiratory problems, ear and eye and skin conditions. And importantly, we're beginning to see also skews in cell composition that are linked to mental and behavioral disorders. So this really opens a possibility of using, as we hoped, single cell data as a molecular readout for disease state. We of course want to move away from a situation where we just can correlate a disease diagnosis with a single cell trait, and we want to be able to dissect cases where indeed the variation in single cell trait, perhaps driven by genetics, by affect or be the cause of disease, from cases when this is simply a readout of a disease state, or indeed cases where the single cell trait and the disease are driven by a common confounding effect. And to do that, we mostly use approaches anchored on genetics, essentially using genetic as an unperturbed representation of single cell states. I don't know if you're familiar with the concept of EQTL and the way we're using in genetics, but essentially the idea is that for any transcriptional readout, in this case derived from single cell, we can map genetic effects and we can identify genetic variants that increase or decrease the expression of a given gene. And we do this because we do this at a single level. We can do this in a completely controlled manner. So for instance, we can identify genetic effects that might affect expression of a given gene in a given cell type, but I have absolutely no effect in other cell types. And this is very powerful because it gives us the resolution to pinpoint, for instance, for disease variants, what is the likely effector causal tissue. So we carried out quite quickly a very comprehensive EQTL mapping effort, looking at the moment only cis EQTL effects, and this turned out to be very, very well powered. So we know from other studies that we expect to see that every gene that we test will have at least an EQTL in them. When we take all genes expressed in a given cell type, when we express the cells as a function of their abundance, we see that about half of them for the most abundant cell type have an EQTL. But indeed, when we are in a position of looking at the highly expressed genes, which are the most well-powered, we see that for the commonest cell types, we have, in fact, absolute power to detect EQTLs. And this is very important because it helps us calibrate not only what are the things that we see, but also the things that we are likely to miss for the samples where we don't yet have sufficient power. And we can model essentially the power to discover EQTLs as a function of the number of donors in the study, the number of cells within the study, or the number of cells per cell type. And what we see is that indeed in this lower end of the of the atlas, so for variants that are pretty rare, we haven't quite yet reached the right coverage to discover every single QTL. At the moment, through this effort of analyzing 6,000 individuals with 28 different cell types, we have over 90,000 genomic regions associated with a SNP is associated with expression in a given cell type. Of this 75,000, they define 75,000 independent associations. Only about 20% are specific. Many are shared between different cell types. And indeed, we can see cases also when a causal variant might be associated with multiple genes. And so this is just some of the scenarios that we're looking at. For instance, we're looking at cases where a genetic variant might affect the expression of a gene in multiple cell types. And so what we see is that if we define pleiotropy models as essentially these models of co-regulated genes, 60% involve more than 2 cell types. We also see many cases, about 35%, where the SNP controls multiple different genes. And so we're beginning to get this really exquisite resolution of transcriptional effects for key genes. So this is just an example of an association at the estrogen receptor where a SNP controls the expression of this gene in all the T cell subsets. But as you see, we can also very clearly see in the cases where the SNP has an effect of increasing expression of the gene versus those where it decreases it. And this is for Piero. This is a long non-coding RNA that has an EQTL that is seen in all 28 cell types tested. It's a highly important long-coding RNA for colorectal cancer. There's a clear subpopulation of cells that behaves in opposite direction to the rest of immune cell types. And while this is early days, what we're beginning really to see is that there is a huge value in this type of data. We localize systematically with disease association to map transcriptional effect to disease variants, and we now have something like 800 independent cis-regulatory modules that are associated with one or more disease risk or risk factor. So some take-home messages from this type of data is that we have developed a robust and computational workflow that enables population-scale single-cell sequencing. We have generated a foundational single-cell resource of PBMCs in 3 different populations. We have identified transcriptional signatures of compositional traits and biological processes and diseases. And we have an exquisite detail of genetic contribution to transcription traits across many, many more cell types, as well as rare cell composition. And this provides insights into disease associations. But we also hope that both the data but also the lessons that we learned in generating this type of data can be of benefit to the HCA community, and really look forward to a greater integration. Of course, this is just really the beginning. We analyze the data very quickly, but we really have a deeper sense of where the value of this data will be fully realized, also as a paradigm for other complex disease. First of all, we know that single-cell effects are not static, they are dynamic. We know about that indeed the population of cells vary, gradients vary, and we beginning to look for genetic effects that, for instance, might alter not so much expression of a gene, but a propensity of a cell to translate into a different cell type. We are incredibly excited about the possibility of using this large data set to begin to map cellular circuits in a population level, so to identify not only through cis and trans-EQTLs, but also through approaches such as dialogue, the possibility of defining clusters of cells that co-regulate in disease settings, or indeed that show evidence of immune crosstalk in different settings. And because the data is so large and there are so many disease diagnoses, we can essentially splice and dice the data in any way we want to understand better these disease associations, but also the impact of exposure on these cellular circuits. We are very keen to understand better what underlies the modes of regulation of this data, and particularly using techniques such as ATAC-seq to allow us to infer chromatin changes that are associated to cell type specific and environmental exposure-driven effects, genetic variants. And so we began to generate single-cell ATAC-seq resources to complement the single-cell data and to address some of these questions. And of course, we really want to push the boundary of how we analyze this data. What are the ways that will better capture the complexity, the circuitry, and the perturbation sides of this data? One is to move from the linear models that I've shown you before to essentially deep learning approaches that allow us to map transcriptional effects and transcriptional cascades from the data, and Dana is taking a lead in this. And also to, again, apply and drive forward the development of foundational models that allow us to search the space of single-cell data using medical records in an agnostic way. So rather than fitting our hypothesis about which disease we see associated with the phenotype, we want to be agnostic and look for essential instances of cell composition differentials that correlate with disease states. And we think this work at the moment is, one of the methods is led by Craig Glastonbury, Human Technopole, but we're working also with other colleagues on that. And so this really excites us because it provides an first opportunity to begin to understand how these fine-grained cellular changes reflect not just in disease diagnosis, but also in disease subtypes and disease progression in our data. And finally, of course, as I mentioned, we're really interested in the genetic aspect, but bearing in mind this thought that this population can be considered as, if you want, a natural perturbation experiment, where every genetic variant, both common and rare, is a perturbation to a system. We want to also really push the analysis of rare variant-driven associations, because we think that individually rare variants would provide a deeper phenotypic effects. And so in Genes and Health, we have a large number of individuals that are homozygous carriers for loss of function mutation or compound heterozygotes, and many of these lead to complete loss of transcript and protein. And so this provides a very exciting opportunity to study the ablation of biological cascades from the single cell. And of course, we also want to use methods that allow us to seamlessly integrate the contribution of every single rare variant to medical phenotypes. And Brian and Eva are trying to pioneer this approach. So I really want to conclude by saying that, I know there's a lot of talk in the community about bringing single cell to population scale. I think this pilot, if you want, shows that the time is really right from a technological and knowledge point of view. We are there to be able to do studies population scale. And I really hope that atlases such as this one, integrated with disease atlases, and this was the topic of the previous talk, really drive forward also the interpretability of studies that are done in more defined disease settings. So we think that this is really going to be a fundamentally transformative resource to understand better in a very fine-grained genetic control of transcription and of cell composition in humans. We think that the transcriptional regulation will provide indeed incredible clues into genetic, phenotypic, and environmental variability. Hopefully that it empowers cell annotation efforts for not only common but also rare types, and give better insights into transcriptional signature of biological processes such as aging and also of diseases. And of course, we are very interested in continuing this effort to annotate the function of genetic variants and develop drug targets from that. I really hope that this resource will be released to the global community by UK Biobank and Genes and Health, and we hope that really will provide a game changer for informing analytical method development and will be used very much at scale. We're thinking about a scale up using the remaining individuals that are still being recalled in the UK Biobank, and really keen to talk to anybody who would be interested in partnering up with us on this. And really, this leads me to acknowledge an incredible team of experts on different fields. So we really were lucky to benefit from bespoke experience in large-scale studies at the Sanger, both by the lab team, but also the software development, R&D, and bioresource components. And I also really want to mention incredible effort by our analytical team that is shared between my group, Sarah Teichmann, Oliver, Fabian Theis, Mark Bonder, and others. And they really push very hard the analysis of this first data in order to be able to produce data for this talk. And finally, also thank you to our funders and the cohort owners. And I'm happy to take any questions. Thank you.
Thank you, Nicole. Wonderful talk. Questions? Yeah, sure.
Nicole, really exciting to see the large-scale correlations you're finding with so many diseases and human diversity as well. That's really a burning question in the field. You identified some cell proportion differences associated with specific diseases, also associated with, say, Pakistani versus Bangladeshi ethnicity. I'm very curious to know, was there any intersection, meaning did the cell proportion traits that were different between ethnicities, were they also disease biomarkers, in which case you'll say the diagnostic cutoffs need to be changed for that disease, depending on the ethnicity?
Yeah, we're not quite yet at the stage where we can answer this question. So we have only linked the phenotype clinical data of Genes and Health and not yet of Biobank. So I think what's becoming quite interesting is that we can model genetic effects, which are inborn from transient perturbation, chronic disease. We're already beginning to see that. But I think we need probably UK Biobank in order to make this cross-difference. What we do know is that because we see that even the disease distribution between the cohorts is quite different. So for instance, Genes and Health has a lot of disorders of pregnancy that are of course not seen or very old in UK Biobank, but we're really at the beginning of this type of journey.
Well, I can make so you've shown that it's very important to sample a certain number of individuals. But if you think at the brain and all the diseases related to all the neurodegeneration, psychological traits, the challenge seems to be absolutely huge because we should sample more and more diverse region. Do you have any possible insight? Is that going to be possible, or shall we think bigger?
Yeah, I think partly, the value of blood is to be able to arrive first to the numbers that are unachievable in other tissues and try to learn some of the question, right? And so, of course, some of the genetic question need a lot of power. But I think atlases like this can provide, if you want, a foundational, if you want, grid onto which to anchor, other types of data. So we know GTEx has done a lot of brain ETLs, for instance, in postmortem samples, but it's quite unclear to what extent you can capture, ongoing event. One of the things we're thinking about that is that, for instance, we can take genetic markers of cell composition traits and then go back a baseline with those genetic markers and then predict the long term of disease. And this should be quite powerful to be able to model what is really a trajectory of disease and what drives it.
Hey, Nicole. Thank you. This is a very nice talk. And I think people have been, talking about this for a long time to do like really population scale single cell sequencing, and you actually pulled this off and with, really fast and tight timelines. So kudos to that. I have I guess from a population genetics point of view, right? GTAC's colocalization only explain about 40% of GWAS loci across 20 to 30 diseases. Yeah. And I'm really happy to see that using single cell data, you increase that number to 72%. I saw on your slides. So previously using GTACs, people think that, because we're not doing cell type specific signals, so some of the cell type specific signals have been masked by homogenization of the cell types. But now we have single cell data and still only explain, well, it's definitely 30% more, but there's still 20% that's not explained. Yeah. Can you speculate on what that remaining 20% is? Is that different molecular phenotypes like splicing, post-translational modifications, or different perturbations? What do you think?
Yeah, and to be clear, we're not yet at the stage where we can conclusively say what we don't know. We see what we see, but what we are beginning to do, there's a lot of evidence, for instance, colocalization happen at secondary rather than primary signals, and and the rate there might be higher. We are certainly powered to look at effects of, because we have combined, for instance, proteomic data in these individuals, we can begin to understand whether other types of regulation, including stability of protein, could explain the remaining loci. But we don't know that yet. But what we do gain from this type of data is the fact that, when we see a colocalization with monocytes, so we worked in Blueprint for a long time, we really don't see the other cell types, and so it's a speculation whether that's a causal cell type or not. And you see here, actually, we can reconstruct fully the co-regulation patterns of all genes, and this really is quite powerful to be able to not just to rule out or include candidate associations.
Okay. There?
Yeah, your wonderful talk. The clonal hematopoiesis is one reason why you may see a difference in PBNC composition. Between your two cohorts, do you see any difference in the clonal hematopoiesis?
It's a great question. And so UK Biobank age is on average 67 years of age, with a range between 55 and 85. So they are bang in the middle of when they will develop clonal heme. The Pakistan and Bangladesh have a very different age distribution. The average age is 40, but we have individuals from 16 all the way to 88. So this is how we can fit these trajectories. So we expect UK Biobank to have a lot of clonal heme, but we have, because we're doing two things. We have clonal heme readouts at baseline 20 years ago, but we don't have them at this time point, and we don't have the money to sequence, although we'd really like to sequence those sites. We are trying other approaches. So, for instance, we're trying to infer clonal heme from the single cell data, but we're not quite there yet. The other, we're looking at the clonal, we know that essentially clonal heme affects mostly the myeloid compartment. Actually, most of the age-associated changes that we see are lymphoid related. So there are changes in the cells that we expect to see. So cytotoxic cells go up, and I think Piero showed that already in centenarians, but we see that actually as a continuous change in proportion. Temporal cells go up. So, there's, I think there seems to be a lot of more age effects in the lymphoid compartment. I think, however, it will be, the moment we have clonal heme readouts, that's really when it empowers also the understanding and the understanding of these effects. The other thing we're thinking about is using genetics. So we have genetic marker for clonal heme predisposition. So even in the absence of a readout for the somatic mutation, we can model whether a person is at the extreme of clonal heme risk from inherited polymorphism. And so we can use that as a proxy of whether a person is more or less likely to have a clonal heme, with the caveat that it's not predictive, but yeah.
Okay. Maybe the last question, Shyam?
All right. Very curious to know, because the eternal question is how much of the expression variation is accounted for by genes and environment. I'll refine that by saying, say, the genetic variants, the ancestry, age, sex, comorbidities, any of the environmental variables you have in your metadata. Like, how much variance is explained by all these things? And which wins in the end?
We don't know yet. I mean, we unlinked the data a month ago. This is all data, so there's a lot to do, but very happy to take ideas of, we'll quantify that.
Thank you.
Thank you.
All right, I'd like to thank Nicole again. And thank you. And I'd like to thank all the speakers today, all the chairs, all the colleagues discussing, leading all the fantastic discussion groups, and of course all the sponsors as well. So, and now we are going to have this. It's 12 hours that we are discussing. We're gonna, have some probably drinks and food outside with the poster view and also interaction with our sponsor. Thank you again. See you tomorrow morning at before 9. Thank you.